Well, welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. So good afternoon and welcome to the August FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. Last month in this seminar, we heard from Associate Professor Kelly Williams and Dr. Lyndall Hendon from Macquarie University, and they took us through their research into the genetic causes of MND. If you missed out on this talk, it's now available on our website, so you can catch up at a convenient time. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note, however, that the chat function is only enabled if you're using the app-based version of Teams and unfortunately doesn't work in the web-based version. Please make sure you introduce yourself and where you're from before asking your question. So now to our speaker from today. Dr. Sam Barton is the head of the Myelin in Health and Disease Laboratory at the Florey Institute in Melbourne. She completed her PhD at the Hudson Institute of Medical Research in 2015, and following that was awarded an NHMRC ARC Dementia Research Development Fellowship to complete postdoctoral work at the University of Edinburgh. Returning to Melbourne, she was recruited to the Florey to continue her work with cutting-edge iPSC techniques to study MND and frontotemporal dementia. She was recently awarded a Rebecca L. Cooper, Al and Val Rossentraus Medical Research Fellowship and has established the Myelin in Health and Disease Lab at the Florey. She joins us today to talk about some of the work her lab has been doing to understand the role of protective myelin sheath in MND pathogenesis. Thanks, Sam. Over to you.
Thank you, Sarah. Okay, well, thank you very much to FightMND for allowing me the opportunity to showcase some of the research that's coming out of our lab today. It's sort of going to be a bit of a journey through my first couple of postdocs and then into my lab today. And it's going to delve really in depth into myelin, so I hope you're all really excited. And I thought I would start, I wasn't sure how Sarah would introduce me, so I thought I'd just give a little overview of who we are and my team is. So this is my lab here, and as Sarah said, we're based at the Florey Institute in Melbourne, and most of my lab does work on the role of myelin in neurodegeneration and mostly in motor neuron disease. We're obviously very fortunate being at the Florey Institute to be able to work alongside and with Brad Turner and his very large MND empire, which is obviously very fortunate for us because of the breadth of techniques and knowledge and model systems available to us to collaborate with. So within my laboratory, we like to use combined model systems to really kind of phenotype the disease pathologies and make sure that they're consistent amongst the different models. So that can kind of give us a little more assurance that what we're finding is actually valid to what's happening in disease. So the three main things that we focus on are human post-mortem tissue, mouse models, and induced pluripotent stem cells from people living with ALS or MND. And so as my talk progresses, I'm going to use these little icons in the slides to kind of help keep it a little coherent so you know kind of what model I'm referring to. But ultimately, we use the combination of these model systems to interrogate what role myelin plays in MND pathogenesis. So I thought with the audience here, I wasn't going to spend too much time on the background, but I didn't know how to just jump straight into myelin biology. So I'll give a little bit of context. Obviously, most of us listening today would be kind of mostly interested in what is leading a healthy motor neuron to degenerate and begin to show dysfunction in people living with MND. And I'm sure, like many of you, we focus on genetically caused ALS, where we know certain genes cause ALS and the pathologies associated with those, that we can then highlight pathways that are dysregulated and see if we can then extrapolate those to the more randomly caused or sporadic ALS cases. So obviously the main pathology that we work with is TDP43, the protein that we know mislocalizes and aggregates in the majority of people living with MND. The most common genetic cause of ALS is the C9orf72 mutation and an expansion mutation within this gene, which also shows TDP pathology, but has its own unique pathologies that are both at a protein and an RNA level. So within our lab, we work on both TDP and C9orf72, as well as SOD1, to try and encompass sort of all of the main pathologies that frequent in MND cases, to then see if we can find pathologies and extrapolate those to sporadic cases. So obviously a lot of the work goes into working on understanding how these pathologies tie into neuronal dysfunction. But I guess what I want to highlight is we know that the surrounding glial cells around motor neurons also exhibit these proteinopathies. So if we believe that these proteins are leading to dysfunction in motor neurons causing their dysfunction, it seems logical to me to assume that these protein and RNA pathologies may also be contributing to dysfunction in these surrounding glial cells. And so the glial cell that we're particularly interested is in it's the myelinating cell called the oligodendrocyte. So in this schematic here, the oligodendrocyte is the purple cell, and you can see the cell body, I'm assuming you can see my mouse, the cell body extends its cytoplasmic process, and it ensheaths the surrounding axon in these concentric loops of this really fatty, dense myelin. And it's very well characterized that myelin is necessary for maintaining action potential propagation along axons. But we also know that oligodendrocytes through this myelin can shuttle many things, including metabolic factors, trophic factors, endosomes, organelles, proteins, lots of things. So we know that oligodendrocytes are critically important for the maintenance of axonal function. And of course, we also know that the majority of motor neurons are myelinated, as well as some of the inhibitory interneurons that then synapse onto these motor neurons. So I do believe that I'm going to convince you by the end that oligodendrocytes are likely to be contributing to motor neuron dysfunction in MND. We're certainly not the first people to think this. So this paper came out from Dwight Burgles and Jeff Rothstein's lab 10 years ago now, where they looked at human post-mortem tissue. So on the left here, they did stain for a histological label for myelin, and you can see in the ALS case this reduction in the myelin signal. And then these Western blots on the right here with the motor cortex at the top and the spinal cord on the bottom. And you can see, looking at two key proteins, MBP and CMP, that make up myelin, there's a reduction in MBP in both the motor cortex and the spinal cord in people living with MND. And then this was taken a step further more recently with silver staining on the left here. You can see these beautiful parallel myelinated fibers in the corpus callosum of the brain, whereas in the ALS case, there's just sort of chaos, really. You lose these parallel fibers and the myelination has lost its really strict kind of morphology in the brain. And then using fractional isotropy, again, which kind of measures the parallelness of myelin, you can see this significant reduction in ALS cases. And these four triangles are actually C9orf72 cases, again, highlighting that myelin seems to be changed in people living with MND. But this kind of brings us back to the overarching question of our lab. It's whether the loss of this myelin is actually just a direct result of neuronal loss, which seems logical. If we know that you lose motor neurons as disease progresses, it seems logical you're also going to lose myelin. However, we question whether there's actually an intrinsic deficit in the oligodendrocyte itself that could also be contributing to this change in myelin. And indeed, taking you back to that Nature Neuroscience paper I referenced a little earlier, when Dwight Burgles and his lab deleted the SOD1 human transgene from oligodendrocytes alone in the SOD1 mouse model, they found that it delayed onset of disease and also extended survival in these mice, implying that if you have healthy oligodendrocytes, this may be sufficient to prolong or extend motor neuron survival in this mouse model. So I bring you back to this schematic here, and I guess these are the three key questions that we're aiming to answer in our lab. And so we're beginning at this point to just try and address these questions overall in MND. So is the oligodendrocyte lineage altered? The production of the precursor cells that give rise to the oligodendrocytes that give rise to the myelin-producing cells? Is their myelination capacity altered and their capacity to myelinate motor neurons? And then is their metabolic support to neurons altered? So we see these as kind of the three key pathways. And we want to build on kind of that more, I guess, superficial analysis of myelin that's occurred so far to see, is the function of oligodendrocytes altered? Following on from that, once we've identified our phenotypes, obviously our goal will be, are these changes driven by the oligodendrocyte? And therefore, could we therapeutically target these pathways to try and rescue these changes and preserve their support of motor neurons? And could this be kind of a viable way of extending the survival of motor neuron function? So I'll first start by covering one of the projects in our lab, which is looking at the oligodendrocyte lineage. So excuse how sort of mediocre this image is, but it tells you what you need to know. So we've got our oligodendrocyte precursor cell here, which is cycling in the cell cycle. It can then leave the cell cycle, become dormant and remain as an OPC, re-enter the cell cycle, and then differentiate into a pre-myelinating oligodendrocyte before maturing into a myelinating oligodendrocyte. So our question is, could this process be altered, and therefore could there be shifts in the proportions of these cells, leading to either a reduction in OPCs, which is a problem in of itself because they do have neuronal support functions themselves, or could the change in this lineage also therefore lead to a decrease in these mature myelinating oligodendrocytes, and therefore lead to a decrease of the capacity to actually have myelin around motor neurons? And so we address this in two models using our iPSCs, or our stem cells, and our mouse models. And so through this talk, I'll generally talk about the stem cell model first because they're more developmental questions, and then I use the mouse model data to show kind of the context of what we've found in a more chronically aged and diseased context. So this is the protocol that we use for our iPSC. So we take our stem cells from people living with ALS, and it undergoes a 7-week patterning process to generate oligodendrocyte precursor cells. We then put it into kind of a pro-oligodendrocyte media, which is this pink bar here. And then after a few weeks, we get pre-myelinating oligodendrocytes. And after 12 weeks, we get myelinating oligodendrocytes. So it's a really cool system that allows us to phenotype cells at different stages of development. So for the context of this part of the project, we've used a SOD1 case, 3 C9orf72, and 2 TARD-BP, and then isogenic controls for most, except for this one line here, to interrogate whether there are differences in the oligodendrocyte lineage. So this is work of a PhD student, Francois, in my lab. So on the left here, he looked at proliferation of OPCs within our organoids, and on the right here, their capacity to differentiate into pre-myelinating oligodendrocytes. For most of the graphs, we've got SOD in red and the isogenic in orange, and then C9orf is in dark blue, and it's each of those individual isogenic controls in a light blue. And what you can see in both the proliferation and the differentiation, that we had no difference across our lines. So developmentally in people with MND, we can quite, I guess from this, confidently say that there doesn't seem to be any issue with the establishment of the oligodendrocyte lineage. But we want to delve into this a little bit deeper. So taking you back to your first year uni, here is the cell cycle. And so what we used was double S-phase labeling. So we put BRDU and EDU, which are thymidine analogues, into the media. And this labels cells that are in S-phase, or the stage where they're replicating the DNA in the cell. And by adding 2 different factors, we could then track the process of the cell through the cell cycle to gauge, is the actual dynamic process of this changed? And so what Francois found was that whilst in the C9orf, we didn't see any differences between our patient lines and our isogenic controls, we actually saw that in our oligodendrocytes with the SOD1 mutation, a significant reduction in the OPCs in the S phase of the cell cycle comparative to their isogenic control. And this is important because this is the stage of the cell cycle where they're synthesizing DNA. And it's a very tightly regulated process, as I'm sure you can appreciate. So any difference is going to have a profound effect on this cell's function. And even more intriguingly, when we look at the total cell cycle length, which is on average about 35 hours, which is on par with what's been found in embryonic tissue and in mouse models. So you can see that we don't have any difference in the cell cycle length despite the change in the S phase. So this is now something that Francois is going to delve into in more detail to see if this is uniform along multiple SOD1 lines and kind of what's driving this change. So we also want to look at the OPCs functionally. So like I said, whilst OPCs contribute to myelinating cells, they also function in of themselves by reacting quickly to neuronal activity. And so this is not a pathway unique to oligodendrocytes. It's been very well characterized in neurons. This is the AMPA receptor, and we know that during development, its second subunit of its 4 is calcium permeable. Through development, it then undergoes RNA editing to then become calcium impermeable. And we know that in motor neurons in MND, they don't undergo this editing and they remain calcium permeable. And this is believed to be kind of an important pathway contributing to neuronal hyperexcitability. So given AMPA receptors are abundantly expressed on OPCs, we wanted to also see this, whether this was affected in our OPCs as well. So on the left here is TDP43 iPSC lines, and on the right is the C9orf. And so what you can see is that it measured effectively the shift in this AMPA receptor. So whilst in our control, our M337V TDP line, you can see that with maturation, you get this reduction in conductance, which is linked with that RNA editing and that shift to calcium impermeability. In this 2 clones of this TDP line that has the G298S mutation, we lose this editing. So the AMPA receptor remains calcium permeable, which is rescued in the isogenic control. Comparatively, in the C9orf 72, we have 2 patient lines here, and you can see that as they mature, we also get this editing. So it does seem to be, in this case, unique to this point mutation. So certainly something for us to follow up as well. So as I said, iPSCs, obviously we're modelling something more developmental at this stage. We know that iPSCs are mostly embryonic in their transcriptome. But I still think they're critically important, firstly because they're human cells, but also because it gives us awareness of what's happening kind of intrinsically to oligodendrocytes, separate from chronic changes in response to neurons. But using complementary mouse models allows us then to look to see what changes occur when you do factor in chronic disease as well as aging. And indeed, in the SOD1 G93A mouse model, this was published by Dwight Burgles in that same paper I keep referencing. This is a lot of information, but effectively I just wanted to show you that in the SOD1 mouse, even from very early on, there are changes to the oligodendrocyte population where they had increased proliferation and increased density of cells overall. And so in our lab, Katie, one of my PhD students, is working on the TDP43Q331K mouse model. And I've just shown you some of her data here, but at end stage, she sees the same thing. So we've got increases in the density of oligodendrocyte precursor cells and all oligodendrocytes. We have increased proliferation as well as increased density of dormant OPCs. Differentiation is increased as well as the density of mature oligodendrocytes. So this does seem to be something that is common amongst both genotypes, which is very exciting to see. So after looking at the oligodendrocyte lineage, we wanted to look at whether the myelination capacity of our cells is altered. And so I wanted to give you kind of a little bit more context. We all obviously are aware of, I hope that everyone is aware of what myelin is, but I wanted to kind of give a little more information. So we know that each individual oligodendrocyte cell body can myelinate up to 50 axons surrounding it. And myelin is actually it's a tightly regulated process where it extends its cytoplasm and encases the axon in these loops, which you can see in this EM image here, these concentric loops of myelin. And they're established in what's called an internode, and then there's a gap left in between the myelin called the node of Ranvier. And it's at this node where there's the high density of sodium channels that allows that ion influx for action potential propagation. So it's both the thickness of the myelin around the axon and the placement of these internodes along the axon that really regulate the capacity for axons to have action potentials and therefore communicate with one another. So I can't really undersell how important it is for this myelin process to really occur properly. The other thing I wanted to mention before I show you the data is this is actually from an entirely different biological paradigm, but the concept is the same. We know that whilst oligodendrocytes are quite rigid throughout life, the production of myelin is very dynamic. So here, in response to an environmental change, there is an addition of myelin added in. So what I want you to appreciate is that in MND, we know neurons have increased activity. They have neuronal hyperexcitability. And so to an oligodendrocyte, this would be seen as a shift in neuronal activity and therefore prompt them to add more myelin. And so again, coming back to the idea that oligodendrocytes themselves are diseased in MND, if they themselves can't myelinate properly, they may not be able to respond to this demand that's being placed on them by neurons. The one last thing I wanted to highlight is that inhibitory interneurons, some forms of them are also myelinated. So again, if the myelin has an issue, then we may impact on this inhibitory interneurons effect of inhibiting motor neuron function. Again, kind of tying all of these pathways together. So from this, we wanted to kind of assess the main characteristics of myelin to see if they altered in MND. So look at internode and node formation along the motor axon, and also look at myelin compaction around the axon and the composition of this myelin. So again, beginning with our iPSC model to see if there's a change in the developmental myelin process. I bring you back to this schematic. And again, this time we're going to focus on this last time point when we have myelinating oligodendrocytes, obviously, so we can now phenotype changes to their morphology. So if I would just zoom in a little bit more, so this organoid model is used quite extensively in our laboratory and also for multiple projects. But it's a really cool system where after a very long time, we get these beautiful organoids that contain kind of all the key cell types of interest for us. So we've got HB9 and CHAP positive motor neurons, as well as parvalbumin positive interneurons, GFAP positive astrocytes, and a lot of them, as well as the entire oligodendrocyte lineage. So it spans from PDGF receptor alpha oligodendrocyte precursor cells through to these MBP positive myelinating cells. And so it's a really kind of a novel system that gives us kind of a large amount of myelin that actually hasn't been modeled previously in vitro. And so here's a tiled max projection image through one of our organoids, and each of these kind of bundles of tubes is an individual oligodendrocyte. So we have the resolution to look at individual oligodendrocytes and their capacity to form these myelin sheaths. We know that this myelin is mature, so in the red is our internode. The green Caspar is the paranodal marker, which then is either side of the node of Rombier, which is the black gap here between the two internodes. And then our myelin is compact, and there are large concentric rings of myelin around our axons. So we believe that this is kind of an ideal model system for us to interrogate myelin morphology in MND. So for this part of the project, we used many more lines. So we've got 6 SOD1 ALS patient iPSC samples with all different point mutations, with 2 of them having an isogenic control. We've now got 5 C9orf72 patient lines, 3 of which have their own individual isogenic controls. We have 1 TARD-BP patient line and 3 unrelated controls to use for those lines that don't have their own CRISPR-Cas9 generator control. So as I said, the brilliance of this system allows us to kind of have that high resolution to hone in on individual oligodendrocytes. So even if we kind of wanted to do this in mice, it is very challenging given the density of myelin in white matter tracts. So these organoids do allow us kind of a really powerful ability to look at oligodendrocytes with single cell resolution. And so these data were collected by Joel in my lab. And so I'm first going to start by showing you the paired lines. We wanted to see, can we identify a phenotype before then extrapolating it, I guess, to more lines. So for these data, Joel counted the number of sheaths that each cell can produce. And so what you can see here in the red bar, which is our SOD1 oligodendrocytes, they actually produce significantly more sheaths per cell than the respective isogenic control. Whereas in our C9orf72 oligodendrocytes, they actually produce significantly fewer sheaths per cell than their isogenic control. So we have this really cool kind of genotype-specific effect between these patient lines. And these data are quite robust. We've done them many times. Joel has done these many times where we've been able to do independent differentiations of each of these lines. So we're very confident with these data. And then inversely, looking at the sheath length per cell, again, done by genotype, you can see that in our SOD1 oligodendrocytes, despite having more sheaths per cell, these sheaths were significantly shorter in our SOD1 compared to the control. And again, despite having fewer sheaths per cell, the C9orf72 oligodendrocytes had longer sheaths per cell comparative to the isogenic control. So keeping in mind the strict necessity of myelin to be maintained along these axons, we are seeing these genotype-specific changes to their morphology. So it'll now be interesting to see kind of what effect this has on function. So now that we've identified a phenotype in our myelin in these organoids, we wanted to then extrapolate that to more lines. So keeping in mind, obviously, with organoids, they have their inherent variability after 5 months in culture, but also we're using human lines, human stem cell lines, so they are hugely variable between each line. So we were excited to find this phenotype to then see if it carried over into more lines. So for these data now, on the left, we've got each individual cell line color-coded by genotype with our unrelated controls merged in the one bar in the black on the left. And then combining them all by genotype on the right, you can see that actually even using our unrelated control lines, we're still preserving this same difference. So our SOD1 lines still have significantly more sheets compared to control, and our C9s have significantly less sheets per cell than the control, with the TAR-BP, keeping in mind it's just one point mutation at this stage, not being different. So I can't really undersell how exciting this is given the amount of variability in our organoids. There's obviously some line-to-line variability, but the fact that we are finding or mirroring these changes using unrelated patient lines that are cultured for 5 months, we're really excited by this. And similarly, we're then looking at the mean sheath length per cell, again drawn out for each individual cell line on the left here. And again, each of these has been differentiated multiple times from iPSCs. And you can see that the trend is still apparent with the SOD, but we've actually lost that statistical significance. But we still do have this significant increase in mean sheath length per cell in our C9 comparative to the control. So now that we've identified these phenotypes, obviously we have a lot of next steps. So the logical next step in my mind is to start adding in sporadic ALS lines. Now that we know that we have these differences between our lines that are unrelated, I'm intrigued to see if we add in sporadic ALS lines, whether we will see a separate grouping in those, or whether if we add in enough, we can actually start to kind of group together some of these lines. It's obviously a large endeavor, but something that we're going to sort of start dappling with to see how we can go. The obvious next step is to look at the functional effect. And so Lee Jun, a PhD student in the lab, is optimizing the use of multi-electrode arrays on our organoid systems. So these are in her organoids that don't have myelin in them. With our C9 on the left here, you can see this kind of hyperexcitable phenotype comparative to its isogenic control on the right. So we're excited to see if we can apply this kind of technology to our organoids to see if we can kind of hone in on differences between myelinated cells versus unmyelinated cells, potentially with single cell resolution rather than the MEA, but then factoring in the MEA to see if we can pull out differences, I guess, between these oligodendrocyte versus neuronal contributions. And then the last thing we wanted to look at is whether these changes are entirely oligodendrocyte driven or neuronally driven. And whilst I think ultimately it doesn't really matter because we're finding phenotypes that mirror what would be happening in a person where all their cells are diseased, I think determining what's driving these changes will allow us to more accurately work out if they are capable of being therapeutically targeted. Can we preserve these pathways, rescue these myelin changes, and will that help motor neurons function? And indeed, from the point of view of changes being oligodendrocyte driven, these are some data from a human post-mortem study that I did in my first postdoc. This is using RNA-scope to look at individual mRNAs of myelin basic protein, which is one of the key proteins of myelin. So each dot is an individual mRNA, and you can see in a control brain, these mRNAs are all throughout what would be myelin sheaths in the motor cortex, comparative to the sporadic ALS cases and the C9orf72 ALS cases, where we saw this huge shift in their morphology that seemed to mirror the pathologies that would be apparent in these cells. So in the sporadic ALS, obviously they would have TDP aggregates that mirror where these MBPs are aggregating. And similarly in the C9orf72 cases, where they also have TDP like here, but they also have these profound RNA aggregations in the nucleus where the RNA foci would be. And so we have quantified this and shown that it's significant, and it implies to us that oligodendrocytes do seem to have intrinsic deficits separate to neuronal impact. But to kind of dive into this in a little more detail, we're going to use comparisons between our organoid models and our mouse models. So these are some more data using our organoids from a line with the TDP43 mutation. This is a whole organoid imaged here with the red being the myelin, and then these zoomed in images of our individual oligodendrocytes between our patient line in the middle here and its isogenic control on the right here. And so similarly to our data that I showed you with the green bar before, the TAR-BP G298S mutation doesn't appear to change the number of sheets per cell nor the mean sheath length per cell. So this is probably not the right example to use to prove this point, but we're now validating this in our lab. But in order to tease out whether the changes that we have observed in the myelin sheath length and number are oligodendrocyte driven, we're going to use a mouse model. We're going to use the Shiver mouse model where they lack myelin basic protein, so they no longer have compact myelin around their axons, comparative to the control where they have this very normal dark myelin around. So what this allows us to do is transplant in iPSC-derived oligodendrocytes. And it means that we're going to get human myelination around the mouse axons, which I know sounds wacky, but it's a really powerful system because it means that we kind of have the same ability to do our analyses now in these mouse brains comparative to our organoids. And if we compare our data between what we get in these transplants comparative to our organoids and they're the same, we can assume then that that's an oligodendrocyte-driven change, because the only difference between these mice is going to be the oligodendrocytes that we transplant in, whereas the axons are going to all be the same. Whereas in our organoids, obviously all the cells harbour that same patient point mutation, and so it's unclear whether the change is coming as a response to neuronal activity or is oligodendrocyte driven. I hope I explained that clearly. But Francois, my PhD student, is now working on this project to add in the C9orf and SOD1 cases, because obviously the TDP was not the right one to start with, but proof of concept, we showed that we can measure internode length in our transplants similar to our organoids to kind of have that comparison between the two. So stay tuned. So again, we then wanted to validate whether we find these changes in the mouse model when we then factor in chronic disease and we're factoring in aging. And so similar to the data before, but I've just given you the kind of breakdown of this phenotyping of this mouse. The TDP43Q331K mouse model is quite a mild phenotype, so it is not a terminal mutation in this mouse, but they do have loss of motor neurons, they've got behavioural phenotypes, and they have neuromuscular denervation and gliosis at this 10-month time point. And so Katie collected mice throughout the span of these mice and then looked at the myelin within them. So to do that, she used score imaging or spectral confocal reflectance imaging. And so what this does really is it takes advantage of the fact that myelin, being very lipid dense, reflects light. And so it allows the confocal microscope to shine its laser onto the myelin, and then its refraction off myelin allows us to kind of pick up these images on the right here. And so complementary to EM, it allows us to look at the compaction of myelin, but with lower resolution, but with a larger area. So it's a really kind of exciting technique that allows us to look at myelin kind of more grossly, I guess, in our areas. So we've got dorsal column on the left here and our ventral grey matter on the right. And so when Katie did these analyses, again, we've got dorsal column on the left and the ventral grey matter on the right. You can see that each of these is presented as a fold change. There seems to be a trend at the beginning where the TDP mice have actually reduced myelin comparative to the wild types. However, by end stage, I mean, this is almost a 250-fold increase in myelin signal in these mice comparative to control. And this is mirrored also in the ventral grey matter. So we do seem to see these changes to myelin in this mouse model. And so obviously Katie added in EM. It's the gold standard when assaying myelin. And so she looked at the G ratio, which is the proportion or the thickness of myelin comparative to axonal caliber. And you can see in the TDP mice, despite this looking quite mild, this is very significant actual effect on myelin thickness in our TDP mice comparative to wild type. And it's what you would routinely see, I guess, in diseases that impact myelin. And so now Katie's going to look at this in more detail to work out, are these changes to myelin, which are unexpected, I would think, having increased myelin in these mice. Is this because there is more myelin around these axons linked to perhaps the changes in neuronal activity, or is the compaction of the myelin altered, and therefore actually would this be kind of more of a disease phenotype? And so finally, we've discussed looking at the myelin morphology and the internode length, but we now want to look at the composition of the myelin. And so keeping in mind that they're concentric loops of this lipid-dense cytoplasmic sheath, each of these is very lipid-dense within the myelin proteins interspersed. And so most studies preceding these have focused on these protein levels. But actually protein only constitutes about 30% of myelin. So we were more interested in characterizing the lipid composition of myelin. And so Gemma was my master's student, And she did targeted lipidomics in the motor cortex white matter from in human post-mortem tissue. The idea of this was using motor cortex, it's where there would be less axonal loss, so the changes may more likely be oligodendrocyte driven. And the idea is that, I guess, any shift in this composition of the myelin may affect its charge or polarity, and so therefore changing the compaction of this myelin, perhaps the positioning of receptors or channels, therefore potentially impacting the oligodendrocyte's capacity to support axons. So this is a heat map showing the top 30 dysregulated lipids. In our sporadic ALS cases, there's 8 of them on the right in green, comparative to the 8 age and sex-matched controls on the left. Keep in mind this is human post-mortem tissue, so this is actually a very decent, I think, heat map showing changes between sporadic ALS and control, despite the obvious variability there would be between human post-mortem samples. And when quantified, you can see that looking at cholesterol esters in our sporadic ALS cases, these were significantly increased uniformly across the different species in our sporadic ALS comparative to control. I've lost my headings, but we've got ceramides on the left and sphingomyelins on the right, and you can see that these are significantly decreased in our sporadic ALS cases comparative to control. So obviously this shift in lipid composition, with myelin being the predominant lipids in motor cortex white matter, we can kind of assume then that myelin composition may be altered in this region. And indeed, Gemma did some correlations between our findings and found that the average amount of cholesterol in our sporadic ALS cases was actually inversely proportional to disease duration, suggesting that the more cholesterol that these patients had in their brains, the worse off their kind of prognosis. And this is being looked at by other people in the lab, and certainly something that intrigues us as well, because we know that cholesterol obviously should be very tightly regulated, but we know also in ALS that the glial cell types have lipid droplets, which would have a lot of cholesterol in them. So sort of looking at the amount of cholesterol and what cell type is kind of responsible for those would be a really interesting next experiment. Okay, so they're the first 2 questions. The last one is sort of at a more functional level. So are these changes that we've noted in the oligodendrocyte lineage and the myelination capacity therefore impairing their ability to functionally support motor axons? So metabolism obviously is a very broad term, but we know that the metabolism of people living with ALS is impaired. So this is mouse data to start with. It's the SOD1G86R mouse. You can see that the body weight of these mice is significantly reduced throughout their lifespan, despite an increased food intake over this same time period. And the energy expenditure in these mice is increased both overall, but as well as at resting, implying that these mice have significantly altered basal metabolism. And indeed, when these mice were fed a high-fat diet in the dashed line, you can see that it shifted their survival curve significantly to the right. And indeed, this is a very small clinical trial, but it was published a while ago now. But in ALS patients that consumed a diet that had either high calorie or high fat, you can see that there was a shift in survival comparative to patients that did not have this extra nourished diet, again implying or supporting the contention that metabolic pathways are severely dysregulated in people with MND. So why is this relevant? We know that astrocytes and oligodendrocytes are responsible for shuttling lactate to motor axons, with lactate being a key energy source for motor neurons. So keeping in mind that motor neurons, both upper and lower, have are very long, and so in order to dynamically respond to the surrounding environment, would heavily rely on these surrounding glia to maintain their metabolic support. And so we therefore question, could there be an impaired ability of oligodendrocytes to metabolically support motor axons, which could be contributing to these kind of metabolic or gross metabolic measurements that have been seen in people and also in this mouse model. So again, we go back to the iPSC model. This time we're looking earlier on at our pre-myelinating oligodendrocytes. This is a very complicated slide, but effectively I just wanted to show you that we have our 2 unrelated controls. We've got 3 CNI-NORF cases again, and they're isogenic controls, and then 2 TARD-BP lines and 1 isogenic control. And I just wanted to show you, even though I've muddled all the colors up, I now realize that they all effectively make beautiful pre-myelinating oligodendrocytes. So to look at their metabolic capacity, we utilized a machine called a seahorse bioanalyzer. And it's this machine on the left here. And effectively, it relies on the concept that when a cell utilizes glucose and undergoes glycolysis to produce pyruvate and then lactate, when it spits out this lactate into the conditioned media, it also secretes this hydrogen ion, which would alter the pH of the conditioned media. So this seahorse machine effectively measures shifts in acidity or pH in the conditioned media as a surrogate for cells' capacity to undergo glycolysis. And I won't go into detail of this pattern here, but effectively remember this pattern, and it's looking at the ability of a cell to undergo glycolysis at rest, but also under a metabolic challenge. So then looking at our individual lines here, you can see our two unrelated controls on the left. What is apparent is that our three C9orf72 derived oligodendrocyte lines have significantly reduced glycolytic capacity compared to the three isogenic controls, but this isn't apparent in our TARD-BP lines. And so then following that trace that I told you to memorize, you can see in our C9orf oligodendrocytes that not only at rest, but also when stressed, they have this significant impairment to metabolize glucose to lactate and secrete this lactate into the media. So this is obviously very exciting data, but it doesn't really tell us what is going wrong. So we know that oligodendrocytes, as well as astrocytes, take up glucose from the surrounding environment through the GLUT1 receptor. They then undergo glycolysis where they metabolize glucose to lactate, and then they shuttle this lactate through MCT1 in oligodendrocytes and MCT4 in astrocytes through MCT2 into axons. And so really this reduction of lactate that was measured by the seahorse could be driven by impairments at either of these steps. So we now want to delve into what's driving this impairment. And so at the moment, we're trying to do Western blots to look at the expression of these receptors, and we're using metabolomics to try and delve into this point too, whether there's a deficiency in glycolysis. So to address this question, we're back to our organoids, and we're looking at our end stage organoids now, where there is myelin, to see is the shift in glycolysis what's contributing to this impairment that we measured in our seahorse. And so to do this, we've added in, or we've supplemented the media, or we've replaced the media rather, with these metabolites that are tagged. So when we add in glucose or glutamine, it's got this extra carbon. And what this means is then when it goes through mass spec, we can look at all the subsequent metabolites and they will maintain this label, so we can actually track how that initial glucose or glutamine is being metabolized by these oligodendrocytes within our organoids. So we're focused on our C9orf72 lines because these are what we identified in the seahorse data. And so we're still working on these data, so I just thought I would throw it up here to show you what we're working with. But effectively, we are looking at different time points using both the glucose and the glutamine, and it allows us to kind of track its utilization through the glycolytic pathway, so we can identify kind of where the limiting factor is, and then look at whether there's enzymes related to that pathway that could be impaired. So we're excited to see where this data takes us. So in conclusion, I guess what I've shown you is a large amount of random data pertaining to the different genotypes at the different stages of the oligodendrocyte lineage. And I guess this is still really important because we need to identify deficits in these cells that are either unique to the genotypes related to them, or whether they're kind of uniform amongst MND and therefore can be extrapolated more easily to the wider sporadic population. And so if we focus firstly on SOD1, here is our kind of control. We've got our motor axon in purple, the oligodendrocyte in orange, and it's produced its myelin sheaths. We've got our OPCs around both becoming the oligodendrocyte and supporting axonal function, and then our astrocytes as well. And we know that with SOD1 mutations, we have this increase in the number of myelin sheaths, but a decrease in their length. And we've also identified changes to the oligodendrocyte lineage where we have increased OPCs that are cycling, their cycling is altered. In the C9orf72, this time we see the opposite, where we have a reduction in the number of myelin sheaths, but an increase in their length. We don't see a change in their lineage, but we do see this change in the capacity for oligodendrocytes to metabolically support motor axons. And then factoring in the TARD-BP, we've found no change to the myelin morphology, but we've found significant changes to the lineage and also the functions of these OPCs. And actually the morphology of the myelin seems to be different from either its amount or its compaction. So I guess at this point, we would still constitute this as preliminary. We're still trying to piece all of the puzzle pieces together to work out which pathways need to be more thoroughly investigated, to work out how can we then therapeutically target these pathways to see if we can preserve motor neuron function. But I guess what I think we have highlighted is that we can't neglect that these surrounding supportive cells themselves may be diseased and therefore are likely contributing to motor neuron dysfunction in MND. And so this is kind of my concluding statement, or the kind of overarching aim of our lab in a way. Could the altered myelin composition or morphology impact on the motor neuron health and function? And so therefore, could myelin and oligodendrocyte function be a therapeutic target in MND? So with that, I'd like to acknowledge this is my lab in the corner here. They all have helped hugely with the data that we've got today. And of course, Brad Turner's lab and everyone that's been affiliated with accruing all of these data. Of course, our funding bodies, FightMND, that's helped support all of our organoid work. And then all of the people that have generously donated their time, donated their skin for our iPSC samples, as well as the human post-mortem tissue. So thank you very much.
Thanks, Dr. Barton, or Sam, for a wonderful presentation there on oligodendrocytes and their impact in MND. We'll open the digital floor to questions. So just raise your hands and we'll let you through to ask your questions. But while we're waiting, I'll take you back to some earlier slides where you were talking about the S phase and synthesizing DNA, and then talking about calcium permeability versus non-permeability and the AMPA receptor. Yes. Have you looked into whether any other receptors are involved in this function in your modelling, or would you like to comment on any roles of other receptors as well?
So the short answer is no, we haven't looked. AMPA receptor was the first one that we looked at because I guess it was already established paradigm in the lab for the people that were already working with neurons. So it was sort of an easy assay to then extrapolate over to the OPCs. We know that the OPC kind of ion homeostasis regulation is very important to supporting motor neuron function. So it's something that I think we should delve into. I think until recently, OPCs weren't really considered as their own cell type. They were always considered as precursors to the myelinating cell. So I think with the more attention they're receiving and our broader understanding of their functions, it's something that we should look at. Yeah, so no, but hopefully soon.
Right. Just waiting for some questions to come up. Can you comment on whether there are examples of myelin dysregulation in other diseases as well, other than motor neuron disease?
Yeah, so I think Parkinson's disease is a pretty cool one to look at, because I think it comes back to the concept of we focus on the cell type that's affected. So in Parkinson's, for example, the dopaminergic neuron, but that's an unmyelinated axon, but the interneurons that would synapse with them would be myelinated. And it's a similar concept in MND where we kind of neglect, I guess, the impact of the inhibitory interneurons. But certainly there are myelin changes in Parkinson's disease. It's also closely linked to multiple systems atrophy, which shares the alpha-synuclein pathology, but it's unique to oligodendrocytes. And I think it's certainly garnering attention. A lot of single cell sequencing data is coming out now, which is sort of accidentally highlighting oligodendrocytes. So Alzheimer's disease, a lot of the single cell data coming out of brain samples is highlighting oligodendrocytes, which people didn't really mean to. So I think that with the improved models and the capacity to model myelination, hopefully now they will gain a little more attention, I guess, in the other diseases.
Are you just looking into the data there in terms of differences between the kind of types of MND, SOD1 versus CNI North, Are you surprised by the kind of differences there, like in terms of increased sheaths per cell, or between the the different types?
I guess it makes it much more complicated, but I don't think surprise per se, because so TDP, I mean, obviously binds to a lot of RNAs in all the different cells, but certainly in oligodendrocytes, it binds to a lot of or it splices a lot of the myelin relevant genes. And so the MND cases that would have TDP issues, I would assume would have different impacts to something like SOD1, where SOD1 isn't as related to oligodendrocyte function. So I guess it's harder then to extrapolate, but I think it makes sense that there would be differences based on genotype.
Just
if we don't have any questions from the floor, we might just leave it there for today. Thank you so much, Sam. If anyone else has any other questions that they would like to follow up with Sam, please do get in touch with her directly, or if you need help getting connected, please contact us at FightMND and we'd be more than happy to connect you. Today's seminar has been recorded and will be available on our website in about a week's time. So if you have any colleagues who weren't able to join live today, just let them know that they will be able to watch it soon. So thank you again for tuning in to the scientific seminar today, and we hope this series has been creating new opportunities for ideas in our research community. The September seminar will be held on Tuesday, September the 26th at 12 p.m. So keep an eye out in your inboxes for details of that coming up shortly. So thank you again, Sam. All the best with your lab's work, and we'll be in touch soon.
Thank you.
Thanks. You're good. Everyone's hanging out. All right. All good?
Thank you, Sarah.
Amazing work, Sam. Amazing work, Sam.
Oh, thanks, Darvor.
Really like the images and the detail, like the precision that you're able to get now with not just the microscopy, but the confocal, and really looking at the changes in the structure and things, before actually delving into the functionality as well.
They just want to know what effect it has on motor neurons. Like, I don't blame them, but it just, the amount of time it takes to get to this point. Want to give the people what they want, but we're just not there yet.
Yeah.
You had to wait for so long to get that differentiation happening, and it doesn't happen overnight for sure.
No, but it's cool. We're starting to add in some drugs now to measure their effects. So hopefully I'll present again next year and there'll be some more dazzling data.
I really like the confocal images you have are really beautiful.
They are really nice.
They're really nice.
Oligodendrocytes. Yeah. Sorry if I took it too long. I thought that I would not have enough, but then I just checked the time and I just waffled on. Sorry about that.
Apparently no one had any questions, so you explained it very well. And it's good that you took up more time then.
Yeah, well done. Well, 3 questions you thought of at the end. I should have given you some to save you the stress.
No stress. I just struggle with asking a meaningful question. That's all.
No, they were good. They were good questions. And
without going into like, with technical detail rather than biological kind of understanding of the research. That it could have gone down the path of, well, so which one do you think? Is it motor neuron driven or is it oligo driven?
Yeah, I'm glad you didn't ask me that.
But
that was my next one before Sarah cut me off. But
you could have done obviously
it anyway. That's a tongue-in-cheek question, obviously, because like you were saying before, it's all relevant to both sides of the equation are relevant because you're getting the different targets that you can aim at. So yeah,
living the dream, guys. We're living the dream.
Living the dream. All right, I'll let you go anyway. Thanks again.
Thank you. Talk to you guys later.
See ya.
See ya.